So in this chapter about the blood, we're going to see what are the functions of the blood, describe and identify the components of the blood, and explain the structure and function of erythrocytes, white blood cells, platelets, and we're going to see how formation of blood cells take place. And as well, we're going to see how we stop bleeding, and we're going to classify blood types according to the antigens that they have, and we're going to differentiate the different types of anemia. So blood is the only connective tissue fluid, and actually the only fluid tissue that we have in our body, and it is transported within the blood vessels, part of the cardiovascular system. And functions of the blood is transporting nutrients, oxygen, CO2, hormones, drugs that the person might take, etc. The blood has a role in regulating the pH as well, because in the erythrocytes we have enzymes like carbonic anhydrase that can absorb hydrogen ions, and then it can help for these to be converted into carbonic acid, and then it will be converting this carbonic acid into bicarbonate and hydrogen ions, and thus can act as a buffer, either absorbing or donating hydrogen ions. The average blood volume in a person, in an adult, is between 4 to 6 liters of blood. Males have more blood than females because their tissues are more denser. They require more oxygen than we, and thus they have more blood volume. And in general, independently of the gender, the blood volume accounts for 8% of the body weight. So then when someone increases its body mass, its body weight, requires more oxygen, the erythrocytes form more, okay? And then the blood volume increases. So there are greater chances then to having high blood pressure with this phenomenon. And then the blood components, the major components will be formed elements, and will be a lipid portion, a matrix that we call plasma. And the blood composition can change rapidly in response to disease. So here are the blood components of a blood sample that it hasn't been centrifuged. So you have a test tube with a blood sample, you see it under the microscope, and this is what you will look. So you will look at cells like these ones that are white blood cells. White blood cells are less abundant and larger than red blood cells. So these are the red blood cells. The red blood cells can be seen here with a clear area because the center of the red blood cells is very thin, and the light of the microscope passes through really easily than here in the periphery. And then the red blood cells are the only cells in our body that they don't have a nucleus. White blood cells, on the other hand, have nucleus. And this nucleus can have a different shape. Like in this case, this is a neutrophil. It has kind of this horseshoe shape. And then this is a lymphocyte. This lymphocyte has a large nucleus, very little cytoplasm. And this is an eosinophil. This one has a bilobed nucleus. Now, these cells, white blood cells, red blood cells, are formed elements, and then we have tiny fragments of cells that we call platelets. The platelets are fragments of a cell that is called a megakaryocyte that lives in the bone marrow. Platelets are specifically made for us for stop bleeding, so they are part of the hemostasis process. And then whatever is not covered by these cells is the fluid matrix, which will be the plasma. So this is a blood smear. Questions? Now, if you centrifuge the sample, so if you put in a machine, it's called a centrifuge, you spin it really rapidly, these components will separate from the blood. So the blood has three major components after centrifugation, which includes erythrocytes, a buffy coat, and plasma. Erythrocytes form 44% of these components. The buffy coat forms 1%, and plasma is 55%. The percentage of erythrocytes is referred as hematocrit. And the erythrocytes goes to the bottom of the test tube because you will have denser cells going to the bottom of the test tube. The white blood cells are less abundant and less denser than the erythrocytes. That's why they stay on top of these erythrocytes, and they form the buffy coat, but not only the leukocytes, but also the platelets. And then plasma, which makes the majority of the blood components, is 55%, will be mainly made by water. And within this water, you have dissolved proteins, plasma proteins of different types. Like we will see some of the components, but for instance, albumin is one of it. And then we have small solutes, which will be like ions, sodium, potassium, calcium. It can be hormones as well. So these are the components of a sample of blood. Something that helps us to understand sometimes health is to know these percentages. Like for instance, the hematocrit can drop in cases of anemia. Or if someone gets dehydrated, if it only loses water, the hematocrit increases because you have less water. So those are the blood components. Now, several studies are done to blood, but one of the most common ones is the complete blood count, or CBC, in which you take a sample of blood, just like as the one that you saw before, and you study the number and characteristics of the blood cells. So it's not only knowing the numbers, but also maybe some of the blood cells are deformed. They might have a different shape, and that can tell you what is wrong. And typically, the labs, they give you a value, like a range, and then you can compare what the patient has to that value and tell whether something's going wrong or not. So erythrocytes are typically reported as number of erythrocytes per milliliter. It's like about 5 million per milliliter. And you can give a percentage. In this case, typically it's like 44–45%. And then the concentration of hemoglobin, it varies. Males have greater number of hemoglobin. Hemoglobin is a protein that is found inside the erythrocytes. And hemoglobin is measured in grams per deciliter. Males have like around 15 grams per deciliter, females like 14, between 12 to 14. If a female is in reproductive stage and it's in the menses cycle, in the menses part of the menstrual cycle, the hemoglobin, is it going to be normal or is it going to be slightly decreased? Slightly decreased. Why? Because during that cycle, that part of the cycle, it loses blood. And then for the erythrocytes, they can report you what is the size, which is typically between 7 to 8 micrometers. The volume is measured in femtoliters, and the concentration of hemoglobin in the cytosol. can tell you the number and the volume of platelets. Kind of a normal number of platelets will be 150,000 per milliliter. And then they can tell you a differential if you ask for it. So it tells you the numbers and types of leukocytes. So these leukocytes are of different types, you will see. These are the white blood cells. And these, when they increase or they decrease, you can tell what is wrong more or less. So here are the components of plasma. Water makes 90% of the plasma volume. Water acts as a solvent to help for dissolve these solutes like ions that we ingest in our diet and transport these solids throughout the body. So for instance, sodium chloride, that salt that we ingest, will be separated into sodium ions and chloride ions, which is going to be positively and negatively charged respectively. They will be carried throughout the body, and then it can enter through the channels of sodium and chloride into these excitable cells, and potassium as well will enter through these channels, and then we can make an action potential with these electrolytes. So once these ions are dissolved, or these salts are dissolved in the plasma, they become an electrolyte, so a carrier of electrical current. And then 9% of the plasma volume is made by plasma proteins, like for instance, albumin. Albumin is a very abundant protein in our blood, and it helps to transport molecules, like drugs, for instance. And this albumin makes the blood colloid osmotic pressure, So this one helps to keep the water volume inside the blood vessels and not out. So it pulls water in with albumin. And then we have immune proteins that are produced by leukocytes. Like, for instance, we have immunoglobulins. So these are antibodies. We have 5 classes. We have IgA, IgG, IgM, IgD, and IgE. These immunoglobulins are produced by type B lymphocytes, which we know as plasma cells. So these are immune proteins, immunoglobulins. The function of the antibodies is to protect our bodies from infections, cancers. And then we have different types of transport proteins that binds and transport hydrophobic compounds throughout the blood. Like for instance, we have transporters for thyroglobulin that transport thyroid hormones, for instance. This is a hormone that needs to be transported. We have transporter also for metals like iron. We have transferrin, for instance. And then we have clothing proteins. We have different types that helps to stop bleeding. We have factor 12, factor 10, etc., that are inactive. Clothing factors are inactive, otherwise we will have blood clots everywhere. And these proteins, and most proteins in our body, so let me write down here. So most proteins of our body are produced by the liver, with few exceptions, like in the case of these immunoglobulins that are produced by the lymphocytes, the B lymphocytes. So then, if clothing proteins that are specifically made to stop bleeding are produced in the liver, do you expect that someone who's an alcoholic will stop bleeding easily? Yes or no? No. Why? Because they have damage in their liver. So they won't be able to produce enough clotting factors. Okay? So they bleed easily. And then we have other solutes like glucose. And the glucose levels can increase or decrease depending on our fat state. So we carry glucose. Glucose is one of the things that is measured in our blood, one of the components. Amino acids, like if someone ingests this morning eggs, these amino acids will show in their blood, right? The amino acid that makes the egg. Okay? Or whatever proteins the person has ingested. Ions, sodium, chloride, your vitamins, if they have copper, zinc, anything, it will show in your blood. And then dissolved gases, oxygen, carbon dioxide. And these dissolved gases are measured with partial pressure of oxygen and partial pressure of CO2. So we have PCO2 and PO2. So this is partial pressure of carbon dioxide and partial pressure of oxygen. This one is typically 104 millimeters of mercury. This one, like 40 millimeters of mercury. And very blood arterial gases tells you the levels of all of this. And then wastes, like we have nitrogenous wastes. That derives from protein catabolism. If someone has high levels of urea, this is known as azotemia. The person will feel sleepy, almost comatose if it's too high. And the measurement for this is blood, urea, nitrogen, also known as BUN. So if someone has increased level of BUN, then the person have problems. This urea is eliminated by the kidneys. So if someone has renal failure, you will have more than likely, if it doesn't have dialysis, it will lead to azotemia. So those are examples of wastes. So now let's look at erythrocytes. So erythrocytes are cells of the, of course, the blood. It's the most abundant type of cell. They have a biconcave disc shape So that means like it has two concavities or two kind of semi-curvatures going internally, or they can be flattened. And that shape specifically gives them large surface to volume ratio. It helps them to absorb a lot of oxygen. So they are basically, the cells specialized to bring oxygen to the cells, but also carry CO2. And that's the reason why they don't have a nucleus, because the nucleus will interfere with this carrying capacity of the cells. So they need to have the space to carry oxygen and CO2. They measure, according to your textbook, between 7.5 7.5 in diameter side to side and 2.5 of width in the actual edge, okay? Because in the center, they are a little bit less in the area where you see the light going through in the microscope. And since they're very small, about 70,000 erythrocytes will fit the head of a pin. They are nucleated cells, they like a nucleus, and most of the organelles, including mitochondria. And they have their cytosol filled with enzymes and 1 billion molecules of hemoglobin. So they are little pouches of hemoglobin. So if you see these erythrocytes sideways, it will look like this. So from here to here, they measure 7.5 micrometers, and in here is 2.5. And then this central part is very thin. So this will allow the diffusion of oxygen really quick. So oxygen comes in really quickly, so it fills in here, and then it goes through here really quickly. That's why they have this shape, to allow diffusion to happen really rapidly. Imagine if it was a cuboidal cell, an erythrocyte, ooh, the oxygen would be like all the distance that I have to go through. So will be slow diffusion versus fast diffusion. Forgive my spelling. So yeah, there's a reason why then they have this shape. Okay? And then hemoglobin, the protein of erythrocytes, is a quaternary structure where you have, in the adult hemoglobin, you have 2 alpha chains and 2 beta chains. And in the middle of these chains, you have a heme group, which contains iron. And this heme binds oxygen. And when it binds oxygen, you call that hemoglobin oxyhemoglobin, or HbO2. And once this hemoglobin releases this oxygen, it will become deoxygenated, so you call this deoxyhemoglobin. Fetal hemoglobin, instead of having 2 beta chains, has gamma. And that makes this hemoglobin to be more avid for oxygen. So it binds oxygen much stronger than the maternal hemoglobin. So why fetuses have that? Well, all they have in their lungs is amniotic fluid. They don't have oxygen, so they have to compete with their mouth for oxygen. So that's why they have a stronger hemoglobin, And that hemoglobin is called HbF, F for fetal. So this is the superior view of an erythrocyte. So has this disc shape. In the center, you have like this little indentation, so that's one concavity, and the bottom you have another concavity. So this is the side view, 2.5 micrometers versus 7.5 side to side. And this center is specialized to allow the diffusion really fast. And everything in the cytosol basically is hemoglobin, enzymes that allows for this oxygen to be transported, and enzymes that helps in the metabolism of the cells. Now, because these cells, they don't have a nucleus and they don't have organelles, they cannot repair. Erythrocytes, once they are broken, they're total. Lifespan of an erythrocyte, they live at 120 days. Erythrocyte is the other name for red blood cells. How many months is 120 days? 4. So they live 4 months. As they age, they start losing flexibility. These cells are very flexible. And their cytoskeletal proteins start deforming, and since they become less flexible, they don't pass as well through the capillaries, and they get trapped in a tissue from the spleen that's called the reticular tissue or reticuloendothelial system, and then they get recycled. This is the quaternary structure of the hemoglobin. You have these beta chains shown in kind of orange, well, yellow color, and these ones beige color, the alpha chains. And this is the heme. Each of the chains have heme. If you magnify the heme, you will see this chemical structure that in the center you have this iron. You can have F2 plus or F3 plus. It's different state of oxidation. When it's having oxygen, this heme emits like well, not emits, but it changes its color from dull red to bright red color. So oxygenated blood looks more red or brighter red than venous blood, which is deoxygenated. So, erythropoiesis. Erythropoiesis is the process of making red blood cells, specifically. It happens in the bone marrow, in the red bone marrow. Yellow marrow stores only fat, but red marrow produces red blood cells. Young animals, young persons have their bone marrow with a lot of cells, stem cells that produces blood. So they would have red marrow. But as someone ages, or an animal ages, these cells are replaced by fat. And at the endings of the bones, at the epiphyses and flat bones, you keep these stem cells that makes blood cells. So in this case, you have a magnification of the femur neck where you have these stem cells. The stem cell specifically that gives rise not only to red blood cells but also white blood cells is called hematopoietic stem cell. And this is a cell that can become any kind of cell. So stem cells, they are pluripotent. That means that they can make different types of cells. So in this case, with certain factors or growth hormones, like colony-stimulating factors or colony-stimulating units, CFQs, you can make this hematopoietic stem cell to make an erythrocyte colony forming unit. So this erythrocyte colony forming unit is going to become an erythrocyte. And as you can see, this cell is large. It has a nucleus, and then it becomes smaller, and it still keeps the nucleus. This erythrocyte colony forming unit, with the presence of hormones like erythropoietin, will keep differentiating into what it will become the future erythrocyte. But first, it will go under some stages like this one that is called proerythroblast. So in this case, you require erythropoietin. Or erythropoietin. So this stimulates the bone marrow to produce red blood cells. Diabetics, when they have end-stage organ damage, like in their kidneys, they will lose the property of making this erythropoietin. One way to tell is their skin. Even if they have white skin, their color is going to be very pale. Because they don't produce erythropoietin, and they cannot stimulate the bone marrow to make red blood cells. Now, this proerythroblast will become an early erythroblast. And in this early erythroblast, you start synthesizing hemoglobin really rapidly. So you start making a lot of hemoglobin, and you start making it so fast that when you go into the next stage, which is a late erythroblast, the nucleus becomes small, and there is no more space for the nucleus, so you eject the nucleus in the late erythroblast, and you eject other organelles. It declutters. The erythrocyte declutters. I don't need a nucleus, I just need a hemoglobin, and get rid of the nucleus and the other organelles. And then, the first released young erythrocyte is going to be called a reticulocyte. In our blood, in our CBC count, we're supposed to have 1% of reticulocytes. And in some diseases where some people, their bone marrow is not functioning, it doesn't produce red blood cells or white blood cells, they can inject hormones to stimulate for instance, EPO, to stimulate the production of red blood cells. They wait like a few days, like 7 or so, because this process takes like 7 days. And if they don't see an increase in reticulocytes, like if they don't see an increase greater than 1%, that means that the bone marrow is not functioning. It's like a money pit. Give money to the wrong person. Yeah, go and get me these things. You keep waiting. Keep waiting, you never get what you ask for. It happens, I have seen it. Anyway, so then these erythrocytes, when they're ready to go, they go through these capillaries in the bone marrow. And look at the name here. It's a sinusoid. So they have large gaps. So the gap, they have to be more or less like around 7.5, so that these red blood cells can go through there. And then they will mature, and then they will carry the oxygen. So that's erythropoiesis. So then what regulates erythropoiesis? Well, it's the amount of oxygen that we have in our blood, the PO2, partial pressure of oxygen. So here we have 104 millimeters of mercury of PO2. That's normal. But if your PO2 drops, then your kidneys will sense that hypoxia. And then they will start forming EPO, erythropoietin. And then that erythropoietin goes into the bloodstream and then into the bone marrow. And then your bone marrow starts producing more red blood cells. Your hematocrit rises. And then the time for erythrocyte maturation decreases, so they mature much faster. And then you will carry more oxygen, so your PO2 returns to normal, and erythropoietin stops producing EPO. I want to say something else about that. Well, this erythropoietin, we have a synthetic version that we have been made with molecular biology or techniques, yes. So if someone has kidney problems, for instance, they can receive shots of erythropoietin because their kidneys, they cannot produce it. It's like insulin. And iron also is necessary to make red blood cells, vitamin C, and amino acids. So it takes some nutrients to make red blood cells. And then after 4 months, when these erythrocytes become old, they go into the spleen. So the spleen, it's a bloody organ that we have in the left upper quadrant, just behind the stomach, a little bit under our ribs. And then, if you magnify a piece of the spleen tissue, so here we have the cells of the spleen, here it's a capillary, so this is a spleen sinusoid. Then your erythrocytes will be trapped, and then you have macrophages that are going to digest these erythrocytes, start breaking it down, and then you will have the release of hemoglobin, and out of hemoglobin, you will take away the heme, and you will be converting it into a pigment that is called bilirubin. And then the iron will be transported by transferrin so that you can make more red blood cells. And then the amino acids from the globin, they are going to also be utilized again so that you can make more red blood cells. So that's the life cycle, or the recycling of the components of the red blood cell. So in black eyes, you have seen this change in the color. And a black eye is nothing else but hemolyzed blood underneath the skin. So initially that black eye looks black, and then starts changing its colors with the days. And that's because these erythrocytes are being recycled by the macrophages in that area of the body where the person was affected. So let's see. if you remember those color changes. So it goes from black color to thankfully you didn't experience that, but let's say that you see somebody, or if you haven't seen it, don't do it to someone. Because next time you will come like a raccoon. I just want to see the change in color and the oxidation of the hemoglobin, the heme. So from black goes to what? Purple. And then stays in purple, my favorite color.
Is it red?
Green. And then normal color of the skin. So yes, I have seen this, but it's kind of like reddish in a way, I guess. But this one, this pigment, or this color comes from biliverdin. And then biliverdin becomes bilirubin. So this yellow color comes from and then the normal is when so that's the cycle then. So this is hemolyzed blood. And then you get the heme converted into biliverdin and then into bilirubin, which are pigments. And then when your macrophages eat up everything, then there is no more need for having this bilirubin in the skin. And one way to eliminate bilirubin is to make it bile salts that will be used by our digestive system to digest lipids.
Is that why people with jaundice turn yellow?
Yes, that's the reason. Excess of bilirubin in their blood. Okay, now we can have abnormal hemoglobin types, like in the case of sickle cell hemoglobin. This is a mutation in the gene for hemoglobin, a single point mutation in which you have a change in amino acids, and this protein can precipitate, and it is non-functional. So it cannot carry oxygen. Like, think about these globin molecules, guys, like little arms that can open and close. That's why they have this quaternary structure. So as they open, they can grab oxygen, and then they keep it tight a little bit while they transport it. once this hemoglobin, that is oxyhemoglobin, reaches a tissue that needs the oxygen, that low pH that is found in that tissue makes this quaternary structure to open a little bit, so then you unload the oxygen. So you load the oxygen, you bring it tight, so you close the globin part, and then you reach the area where you need the oxygen with this excess of hydrogen ions or increased pH, or well, decreased pH. You know that there it needs to be unloaded. You open up this quaternary structure, you unload the oxygen, and then you start the cycle again. You pick up CO2. So think about if it was an Uber. The hemoglobin of oxygen and CO2 is never empty. It's always carrying passengers. Oxygen, CO2, oxygen, CO2. And never both at the same time. So then in this case, you have a single copy of the defective gene for sickle cell trait versus when you have two copies mutated. This will be sickle cell disease. When you have only one copy defective of this gene, you will have no symptoms typically, and that protects against malaria, which is a disease produced by a protozoan. And this is transmitted by the bite of a mosquito that carries this protozoan. It is found in tropical regions, malaria. The reason for this protection is because the receptor for malaria is absent when the person has this defective gene in a single copy. Now, if the person has two copies of this gene defective, the person will develop something that is called sickle cell disease, and it will have hemoglobin S or HbS. And then, under situations where the oxygen levels drop, like if the person is under so much stress, or if he takes certain antibiotics that can decrease the levels of oxygen, then that poor oxygenation makes the erythrocytes to look like an instrument that is used in agriculture. That, I mean, I won't compare myself to you in age, but I'm fairly young and I never have seen one of these instruments, but I kind of seen it in textbooks once. This is a sickle. So this instrument is used in agriculture. So this is the handle. This is a knife, curved knife, that is used for people to grab grains of wheat, and then they cut the stem of the plant. So these erythrocytes are called sickle cell because they look like a sickle. And because this shape is not this shape that can bend, or they can go through the capillaries really easily, that lack of flexibility and that sickle shape makes the erythrocytes to be trapped really easily in the spleen, and the person will have anemia. So this is an example of sickle cell. This is an scanning electron micrograph, so it's a really nice type of microscope that shows this image of normal erythrocytes and some sickled erythrocytes, one here and another one in there. Problem in here is that if many of these capillaries for tissues like the heart, the lungs, the brain get occluded with that, the person will have several infarctions and more than likely death. So, any questions? Now, for production of white blood cells, leukopiosis. So we have same cell, hematopoietic stem cell in the bone marrow, and then this hematopoietic stem cell can take two directions. It can make a myeloid cell line, or it can make a lymphoid cell line. The lymphoid cell line, it will only produce lymphocytes. So it will become a lymphoblast. Remember, in biology, the meaning blast means immature. And I guess, think about it this way. During our childhood years, we were immature, I mean, because that's part of nature. We start learning and then we know what dangers to avoid and what not. But we were all blessed. So just add the ending blast to your name. So I was a Sulit blast, and then I matured and become a Sulit. Or I was a tomato blast. Yes? And don't stay a blast. So then this lymphoid cell line or lymphoid stem cell will become a lymphoblast, and then it will become a pro-lymphocyte or a lymphocyte cell before the mature lymphocytes. So this will be kind of like the adolescent years of the cells. And then these pro-lymphocytes can either become T lymphocytes or B lymphocytes. And these two are part of the immune system. So now for the myeloid cell line, it can take two routes, myeloblast or monoblast. And the monoblast will become a promonocyte, and that promonocyte is going to be a monocyte, which when it's in the blood vessel is going to be called monocyte, but when the monocyte exits the blood vessel, will become a macrophage. So it becomes a big eater cell. And then you have the myeloblast line. This will become a promyelocyte, and then from this promyelocyte, you will form a neutrophil band, a eosinophil band, and a basophil band. So these bands, which are immature white blood cells, just like the the one that is 1%. You tell me a name, you have a name immediately. You don't have to come again. Reticulocytes. You missed that. I was serious. You will still come, you know. So these are like the version of the reticulocytes. The bands are immature cells. So in this case, we have out of a neutrophil band, a neutrophil, out of an eosinophil band, a eosinophil, and out of a basophil band, a basophil. So these are the mature versions then of these white blood cells, and then there are less mature versions. And same as for the erythrocytes, you have to have colony-stimulating hormones that are specific that makes them to mature into that particular type. So then you have precursor immature leukocytes, committed cells, and stem cells here, and then mature cells here. So differences between white blood cells and red blood cells, well, they are larger than erythrocytes with prominent nuclei. They have organelles, and they do have metabolic reactions that help for them to repair, which it doesn't happen in the white blood cell, in the red blood cells. And then they use the blood as a transport vehicle to go into areas of injury. So one way for the white blood cells to know where they're supposed to go to repair something is by chemotaxis. So when our tissues are injured, they release chemicals. And through these chemicals, these leukocytes, from the center of the blood vessel where they are going, they will start going to the periphery. Let's say that this is the blood vessel, right? And this is one of the walls of the blood vessel, this is the other wall. So I'm a leukocyte. And well, I see that there is a point of injury. So that green light is my chemotaxis. So I'm going rapidly, and then I see that green light, and then I start doing this, and I start rolling over. And then here there is going to be a gap, and then I go in between the cells through a process that's called diapedesis. So they squeeze in between, and then they start repairing. If it's an infection, the neutrophils start fighting the infection. So then we have two types of white blood cells. We have granulocytes and agranulocytes. Granulocytes, they have granules, they have enzymes that help to destroy bacteria, microorganisms, and they are released when they are active. We have these granulocytes. We have neutrophils, basophils, and eosinophils. And then agranulocytes are lymphocytes and monocytes. Mono. So they are called agranulocytes because A means without granules. So we have millions of erythrocytes per microliter of blood normally, and we have between 10,000 to 14,000 per microliter of blood of leukocytes. So why for leukocytes we have thousands, and why for erythrocytes we have millions, like 5 million per cell?
Granulocytes would be more specific?
Let me tell you the general, yes, they are. Let me tell you the general function of leukocytes. Fight against infections and fight against cancers or disease. And then erythrocytes is delivering oxygen and CO2. So why we have more erythrocytes, millions of erythrocytes versus thousands of leukocytes?
Need a lot of oxygen?
Yes, because we need more oxygen and we are not infected all the time. So that's why there's a huge difference. Remember that principle in anatomy physiology, the structure function? So in here will be number function. You have more erythrocytes, well, it's because we need more oxygen than fight infections, right? So here is the characteristics, microscopic characteristics of all of these cells. So first let's start with the granulocytes. So this is a neutrophil. It's again a spherical shaped cell. It has a nucleus with 3 to 5 lobes. So in this case it's 1, 2, 3. They are interconnected. And they have cytoplasmic granules. In these granules, they have enzymes, and they are capable of producing oxygen species, radical oxygen species that can kill microorganisms. And then here we have an eosinophil that it has 2 lobes and has abundant granules in its cytoplasm, and these granules stain red. Basophils, the nucleus is S-shaped, and it has so much granules and these granules stain dark purple, and these granules prevents the nucleus to be seen. So these are the three granulocytes. Granulocytes, like the lymphocytes, are easily to be recognized because they have a large nucleus and the cytoplasm is very thin. This is a lymphocyte that it could be an NK cell, it can be a T cell or a B cell. And when these become active, let's say that it's a B cell or a plasma cell, and it starts producing antibodies, the ratio of nucleus to cytoplasm decreases. So the nucleus becomes smaller, and then the cytoplasm increases because you enlarge the rough endoplasmic reticulum to make more antibodies. That's where you make the antibodies. And then the monocyte, the second granulocyte, it has an U-shaped nucleus, and it's the most large white blood cell that there is. The cytoplasm looks pale blue in color or purple when it's stained. And this one is called monocyte when it's inside the blood vessels, but when it's exiting the blood vessels, it's going to be called a macrophage. And depending on where this macrophage, sometimes it can take a name. Like if it's a macrophage in the brain, it will be microglia. If it's in the lungs, it's going to be called dust cells. If it's in the liver, it's going to be Kupffer cell. If it's in the skin, it's going to be dendritic cell. So sometimes they can have specific names. So we will leave it up to there.